All right, everybody. So what I want to go over with you today is, well, two things we want to look at. The first one I'm going to look at with you is what are your early indicators for dysglycemia and how do you use the micronutrient test to pick this up? A lot of you I spend a lot of time on the phone with. We kind of chit chat. You guys send me a case. We talk about it. And I don't really ever have the opportunity to fully go through the background of why I'm saying some of the things that I'm saying to you. So hopefully as we go through this presentation, you'll get that. You'll kind of understand the thought process that goes into some of the recommendations that I give to you sometimes to translate that back to your patients. But here's the other thing, and I didn't take the time to put statistics into this presentation, but everybody in here knows that there's a rise in diabetes. That's no mystery to anybody in here. But here's what I want you to see, and this is what I want you to think about as we go through this. Patients don't come in, they don't start as having normal blood sugar and then all of a sudden have diabetes or even hypoglycemia or anything else. There's a continuum that has to occur through that state of dysfunction before they can officially be diagnosed with said condition, right? So what we want to be able to do is catch the patients when they're in this gray area, when they're telling us, hey, I don't feel just right, maybe I don't have energy, maybe it's the way I feel surrounding how I eat. We want to be able to correlate that with the labs and then correlate the lab, what we're seeing in the labs, to what the literature says so that we can actually have a basis for the follow-up recommendation that we make, okay? So if you're more of a science person, which I tend to be, I want to see some facts, things like that. If you're more of that person, I got stuff in here for you. If you don't care about that stuff and you're just like, give me the bottom line, I got the illustrations in here to give you the bottom line on the end of this stuff too, okay? So hopefully we can kind of meet everybody's needs here. So let's just jump right in. So let's define dysglycemia and what we're really talking about. And there's two different ways to look at this, and they're handled differently. You've got your hypoglycemia and you've got your hyperglycemia. Hyperglycemia has got this, that's the continuum I was talking about. That's the greater portion of the continuum, okay? You got the insulin resistance, you got the pre-diabetes, you got the actual diabetes in there, okay? And the fact is that most of the patients that we're seeing have some degree of dysglycemia when they're walking in the door. Those of you that I have...
I'm sorry, there's two presentations in your folder. It's the second presentation.
Those of you that I have worked with, you know, when we're talking, you guys hear me say, well, what about the patient's blood sugar? What's your glucose? What's your insulin? You know, what are these markers look like? Because if you're not paying attention to those markers, in summary, those are creating a state of inflammation. And as you know, inflammation is basically at the basis of everything dysfunctional. Every chronic disease nearly has inflammation at its basis at some point, okay? Now, more importantly, why do we need to know this? Well, look at the titles on some of these studies that I pulled real quickly, and let's just look at how broad in spectrum the state of blood sugar dysfunction can be. So pre-diabetes, brain aging, and cognition. Now look at this. It didn't say diabetes definitively. It said pre-diabetes. We don't have to get to 126 on our glucose and above for this stuff to start taking place. All right? We've got evidence here. And something else I want you to pay attention to as I'm going through these presentations, look at the dates on the studies. Not all of them, but the large majority of the dates on these studies are within the last 6 months. Most of them are within the last 3 months. So this is information that's continually emerging out there to build the case of what we do with our patients. Insulin resistance, and look at this, metabolic cognitive syndrome. Again, it's not necessarily diabetes cognitive syndrome. It's not at the worst, you know, at the far end of the spectrum here, but yet this is something that's seen so prevalently now that it's got a name attached to it. Increased cellular and circulating biomarkers of oxidative stress and nascent metabolic syndrome. So hasn't been necessarily identified yet in the labs. Okay. And then aging, metabolic syndrome, and the heart. Now, here's how you start this whole process with your patients. And in your conversation with them, where you want to go, these are the things that you want to ask. These are your kind of your red flags if one of these pop up. So with your hypoglycemics, they easily become lightheaded, irritable when not eating, shaking tremors, This nausea in the morning and they don't feel like eating, how many of you guys have seen that with your patients? You tell a patient, look, you got to eat protein in the morning. I don't feel like eating protein in the morning. Red flag right away. There's some state of hypoglycemia probably occurring there. And then cravings for sweets mid-afternoon, right? So they kind of hit that lull in between in between their two major meals there, the last two major meals. So they need some glucose to pick them back up. If it gets real bad, they'll start showing that in the morning too. And then you go to the other side of the spectrum where you're looking at insulin resistance. And the first one that I put up here, I put this as the first one for a very specific reason. If you ask somebody, do you get tired after you eat? That is a huge indicator that they've got insulin resistance, and you have got to follow up with their labs and figure out why they're having that. The other one that's right in there is they sit down, they eat a meal, and they feel, instead of feeling satiated, after they've just eaten a full meal, they're ready for some snacks, they're ready to keep going. Their satiety signals aren't working for them. Okay? So those are all insulin resistance. You guys can read the rest of those. The rest of those are pretty common ones that you already know. Now, here is what I think is the beauty of doing the micronutrient testing. And this is one of the things that's actually not really talked about that much at all with regards to it, is you'll notice on the left-hand side here, I've got diagnostic. So these are the things that we typically think about. If you got some form of dysglycemia, your blood glucose insulin interaction is probably off to some degree. We know that chromium's tied in there. We know that the fructose sensitivity. I mean, Spectra even puts it in a nice little carbohydrate metabolism block for us. We don't even have to think much about it, right? We know it's there. So that's all diagnostic. We know what we're going to do if we see one of those pop up. But there's this whole other category of dysglycemic markers that aren't labeled as dysglycemic markers that are predictive in nature. And as we're going through this, I'm going to show you where in the studies they're actually indicating these as early predictive markers that if you don't do something about these, the patient has an extremely high likelihood that they're going to develop some state of, whether it's prediabetes, insulin resistance, or all the way into diabetes. Okay. So we're going to talk about each one of these. So let's just do a quick review here on our glucose-insulin interaction, because this is more or less what it centers around, right? So I want to make sure that we all have a fairly good understanding about this, because I think there's some things that we tend just in general conversation to leave out. Most of the time what we'll say is you've got insulin interacts on the cell, and then it allows glucose to be uptaken by the cell. Let's get a little bit more specific about that so we can actually see how these nutrients realistically are causing an impact on these cells. So what you're going to see is that you've got insulin, and insulin does interact on the insulin receptors. Fine, we all know that, right? However, what is generally less talked about is that this insulin receptor and this glucose, the glucose uptake transporter, are not right there together. What you've got to have happen is that this insulin receptor has to go through a secondary messenger system to stimulate these GLUT receptors, these transporter systems, to then allow glucose to be taken up. So these are individual things. So we need to treat them in our thought process as individual things. Okay? Now, to start that off, let's look at the glucose tolerance factor. Our glucose tolerance factor is going to interact directly on these insulin receptors. So to activate that insulin receptor, you've got to have your B3 and your chromium there. Those have got to work together. So it's not just looking at that presentation in that carbohydrate metabolism section and only seeing chromium as your primary indicator for what the status of these insulin receptors are doing. All right? It's a little bit bigger than that. You've got to look at what that glucose tolerance complex is made up of, and it's made up of the chromium and the B3, the niacin there. And here's your validation for that. This is actually one of the older studies. But you'll see glucose tolerance factor, a complex of chromium and nicotinic acid. And you can read the rest of this, but more or less what this study goes on to say, that if you've got someone who is non-responsive to chromium supplementation, if you add D3 into their protocol, you're probably going to see an increased response from the chromium supplementation. That's basically what they found in this. So here's your clinical scenario. You got a normal chromium, then you're looking at the micronutrient test, right? You've got a normal chromium, an insufficient glucose-insulin interaction. Where's the problem at?
B3.
B3, right? Pretty easy. Okay. So it's with your niacin. Now, so if your B3 is low, then you're likely predicting an increased occurrence of the development of dysglycemia. Does that make sense? Because you're beginning to challenge the ability of those insulin receptors to function appropriately because of impairment of your glucose tolerance factor.
Can I ask a question?
Can we hold them?
Sure.
Can you write it down to make sure we get them?
Okay.
So, with your looking at the nutrients that help regulate your insulin production or insulin function, let's not just go with production here, it's overall function. You kind of break these down into your vitamins and minerals, and we're going to take both of these and go through exactly how all these have a role. So let's start out with zinc. What you're going to see in this diagram is that's your zinc molecule. And what I want you to notice is that when you're looking at this, everything kind of filters into the center here. This center, this zinc molecule or this insulin molecule is zinc. So if you don't have adequate zinc, you're not building insulin molecules as efficiently as you need to, right? Not to mention all the other metabolic enzymes that are dependent on zinc in this process. And just to give you a little bit more background on insulin, insulin's a peptide hormone. So it's a protein-based hormone. So what you're seeing in this diagram as far as these strands out here, these are strands of amino acids. Right? So zinc is involved in your synthesis, storage, and your release of insulin. So your pancreas is going to be heavily dependent on the availability of zinc. And a deficiency predisposes you to glucose intolerance and insulin resistance. Now, again, if you like your validation, the results of this study show that the mean value of zinc, copper, and chromium were significantly lower in the serum of patients with diabetes as compared to control subjects. So these patients are going to have a decreased trend in those minerals. So you want to make sure that you're identifying those and giving these patients the ability to overcome whatever their dysglycemic state is by repleting those nutrients. Let's look at magnesium. I think this is a pretty interesting one because you'll notice on the left-hand column here I put down in the lower section, there really hasn't been a defined mechanism of action on exactly what the role of magnesium is in this process, but we know it's there. And in fact, this magnesium research, we got an entire journal related to just magnesium research. So what they found in this is that you'll look at the last portion here. The body of evidence from epidemiological studies consistently shows an inverse relationship between dietary magnesium and the risk of developing type 2 diabetes. So again, it's that predictive nature that we're looking for. We want to be able to have markers to identify early on to say, look, if you don't do something about this marker, this is a possibility of where you could end up at. Now, in kind of digging through this, here's what I found out. And this is a possible mechanism of how magnesium might have a role here. And I didn't find a study that directly linked these things together, but if you kind of put 1 and 2 together, this is what you end up with. So our data indicate that short telomeres, so you guys are familiar with the telomeres, you've got your chromosomes, that kind of egg-shaped pattern within the cells, has our DNA. On the tips of those are where your telomeres are going to be. There's a diagram on the next slide. But short telomeres can affect beta cell metabolism even in the presence of intact beta cell number. So you can have, the pancreas can have sufficient numbers of beta cells, but yet those beta cells still not be working efficiently as they need to because the change in the telomeres is changing the genetic transcription of the proteins to those cells. Okay, it's kind of a mouthful. But basically what you're looking at here, thus identifying a novel mechanism of telomere-mediated disease. So with those telomeres not being as healthy as they should be, or to the length that they should be, really, the patient can be predisposed to the development of some type of chronic disease, in this case, Dysglycemia. And what you're seeing here is that literally the length of those telomeres is a determinant of beta cell function and diabetes pathogenesis. Now, what does magnesium have to do with any of that, right? Because that's what it comes back around to, and that's what's important. So here's what magnesium has to do. So these caps on the end of the chromosomes, those are your telomeres. And what we know about these is that there's an enzyme called telomerase. Telomerase is what keeps these telomeres as long as possible. And again, Longer is better. This is where we are able to correlate biological age versus chronological age. What we know is that telomerase enzyme is a magnesium-dependent enzyme. So if these patients don't have adequate magnesium status, they're going to run the risk of shortening those telomeres because there's nothing to continuously repair those telomeres to keep them elongated. Again, longer telomeres, better beta cell function. There's a couple references there.
Now, if you wear them out, are they reversible?
No.
No.
Yeah. Look at the findings in this study. This was a large study. So you guys are familiar with hearing about things like the Framingham study, these huge studies. This was a study. It wasn't on the order or magnitude of that particular study, but these were the findings in that. Our findings suggest a significant inverse association between dietary magnesium, metabolic syndrome, and its components. Again, so we're not waiting until they've absolutely got the definitive diagnosis of a blood glucose over 126. We want to see prior to that stage. Here's another one for you, just kind of reinforcing the synergistic nature of how these minerals interact with each other. What you're going to find is that impaired metabolism of these elements, so again at the top, your copper, your magnesium, your zinc, may contribute to progression of diabetes and diabetic complications. Now, let's jump over to the vitamins here. So your vitamin D, everybody's checking everybody for vitamin D, right? And is everybody coming up sufficient in vitamin D when we generally check them the first time? No, they're all coming up low. Right? So why is that important as it relates specifically to regulation of glucose? Well, if you look at vitamin D really serves a couple of purposes. What it's going to do, the first thing it's going to do is it's going to affect calcium metabolism. Now, the way that vitamin D affects calcium is that your vitamin D is needed for production of it's a protein, but it's called calbinin. And basically what calbinin does is it uptakes calcium from the gastrointestinal tract. But it's a vitamin D dependent protein structure, right? That's why if you look at it, and I'll show you a follow-up example on this in a case study. This is why if you look at the micronutrient test and you're seeing that the patient has a calcium deficiency, you really need to begin to think about, well, what's their vitamin D status? More times than not, you'll be able to fix that calcium deficiency just by introducing vitamin D with these patients. But in regards to the pancreas and these beta cells, the role of calcium is going to be, it's going to allow the calcium signaling. I think about calcium as being the key that goes in the ignition of your vehicle. So without that key, you can't turn the ignition to fire the processes up that vehicle is supposed to do, right? Calcium is more or less the same thing in our bodies. It's what stimulates these enzymatic processes. It's an intracellular signal. It tells other functions to proceed along, okay? So to allow that to happen in these beta cells, you got to have vitamin D there to be able to do that. All right, so that's the first thing. The next thing is inflammation. We mentioned earlier that, well, I didn't mention this. So here's two terms you want to think about: glucotoxic and insulinotoxic. Those are two terms that are used in the literature, and what those mean is that if you have higher levels of glucose above what you should have, it's creating an inflammatory response. If you have higher levels of insulin above what you should have, it's creating an inflammatory response. The more inflammation that's created, the more damage you have on these beta cells. If you're and what we're doing is we're thinking about the progression of this process. If you're damaging these beta cells, how fast are you progressing to full-blown insulin-resistant type B or insulin-dependent diabetes? Pretty quick, because you're basically impairing your body's ability to produce what insulin it can. Now, what vitamin D does in this scenario is the inflammatory process that takes place related to that. Vitamin D actually has a protective role on these beta cells. It inhibits or it prolongs the apoptotic process that these cells undergo. So it makes them more resistant to this inflammatory cascade that gets kicked off by either too high of glucose, too high of insulin, or a combination, and your other markers that tend to follow in with that. So I want to point out two things on this slide. One, the strengthening link between vitamin D and diabetes, and look at this, it does not differentiate between type 1 and type 2. The mechanisms are different between 1 and 2, and the way that the vitamin D acts here. In type 1, you're going to have more of the influence on your T regulatory cells. In type 2, you're going to have more of the mechanisms that we just went over in the previous slide. Now, here's the other thing I want you to show, or I want to show you. Look at the last sentence in this last paragraph. More specifically, vitamin D to modulate immune responses is of particular interest for both the therapy and prevention of diabetes. Again, prevention, 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 okay? Look at the title of this. Will vitamin D become a new anti-diabetic? So, I mean, does that say something in and of itself in just a title and how strong of a therapeutic agent we have in being able to recommend vitamin D? That's pretty strong. A major deficiency of vitamin D in diabetes mellitus type 2 appears to be an independent factor able to predict an increased risk for future cardiovascular mortality. So, biotin. These next two, biotin and lipoic acid, these are pretty straightforward in what the effects are. Biotin, the way that biotin works in this entire process are through what are known as decarboxylase enzymes. If you don't have biotin available, you don't promote or you don't allow decarboxylation to take place, and that's needed to help metabolize glucose and insulin. But here's what it boils down to. Our results suggest that biotin deficiency promotes hyperglycemic mechanisms such as increased glucagon concentration and decreased insulin secretion and a sensitivity to compensate for reduced blood glucose concentrations. So in a nutshell, here's what that says. If you don't have adequate biotin, your body's going to be more prone to kicking in glucose, kicking in glucagon, which is going to put more glucose into your bloodstream, not a state you want. And then the effects of insulin are going to be less impactful as far as pulling glucose out of your bloodstream. Does that make sense? In result, you're more likely to have glucose in the blood at higher levels under a biotin deficient state. Okay?
Could you do that again?
Sure. We talked about insulin earlier, right? Insulin takes glucose out of the blood and shuttles it into the cell. Glucagon is the antagonist of insulin. So instead of putting things into the cell, it takes them back out. A lot of us have talked about the effects of cortisol. I'll mention this coming up, but one of the primary roles of cortisol is to act as a glucocorticoid. So it takes stored glycogen, your stored blood sugar, or not blood sugar, but your stored sugar, and it puts it back into a usable form so it can be mobilized by the cells, right? It uses energy. Glucagon works much along those same mechanisms. What's going to happen under a biotin deficient state when you have these B8 deficiencies is that you're going to be more prone to the effects of glucagon putting glucose back into the bloodstream, and the effects of insulin are going to be less impactful. Your insulin receptor sensitivity, your ability to uptake that glucose by the action of insulin and pull it into the cell is going to be less efficient. So you've got two things working here. You can't get the blood glucose out of the blood, and you've got a higher propensity to putting it back into the blood. Right? Definitely not the scenario you want. And that's just a simple biotin deficiency. If you noticed, and I did this for a very specific reason, the rise in neurodegenerative conditions in the country, Alzheimer's, Parkinson's, and those are the two big ones that we mostly think about, but there's a lot of others. The rise in those conditions is pretty prevalent. We're definitely seeing an increase in those pathological states. It's probably not really surprising that you're also seeing an increase in your dysglycemic states across the country and across the world as well, right? So there's probably some parallel in that. So do we have any evidence to really substantiate that? And we do, and that's why very early on, when I was going through the initial slides and I said, hey, look, these are the reasons that you need to know about this for your patients, is most of the things that I put up there were cognitive in nature, because that's one of the first areas that patients are going to start to feel it at. I mean, even just think about it from a hypoglycemic standpoint. If a patient says, well, I get mid-afternoon and I get into this brain fog, That's cognitive, right? So it's one of the early indicators that we want to see. Or even if you just ask your patients, I'll show you this in a case study. Memory, noticing a little bit of slip in memory, just not being able to recall those names quite as quick. Little stuff like that. What we want to do is we want to be able to protect that neurological tissue as much as possible. And this study was specifically related to peripheral tissue, and it's talking about these Schwann cells, but this same process also occurs in your central nervous system as well, right? So look at what they found in this. They differentiated between intermittent high glucose and high glucose. Now, of those two, which inherently would you probably think is worse? Consistent high glucose, right? It's not. Having fluctuations in your glucose is actually worse for you than having consistently high glucose, according to the results of this, as far as damaging of your neurological tissue. And again, this is very peripheral in nature, again, looking at your peripheral nervous system, but you can find the same information relating to the central nervous system. And if you're interested in what to look for to follow up on this, whereas up here, when you look at the myelin around the tissues that produce that protective layer, the Schwann cells are what do that in your peripheral nervous system. Your oligodendrocytes are what do that in your central nervous system. So if you're looking to cross-reference some of this stuff for central stuff, oligodendrocytes is the term you want. But look at the final conclusion on this. Treatment with alpha-lipoic acid inhibited intermittent high glucose and high glucose induced oxidative stress and apoptosis in these Schwann cells. Right? So you're not only promoting the inhibition of this inflammatory process, but you're also protecting this tissue and preventing them from dying as well. Now, Spectrox. How many of you look at that glucose metabolism section and then you look right next to it and you go, what is that Spectrox and how is that Spectrox directly impacting what I'm seeing in this glucose metabolism section? The two are directly correlated with each other. They're peas in a pod. Okay? So, what you should start to think about when you're looking at that Spectrox marker is the lower that Spectrox marker is going, the higher that patient's inflammatory status is. Those two are directly inversely related to each other. So recent studies have shown that type 2 diabetes patients have increased systemic inflammation and that this state can induce beta cell dysfunction and death. So what did we talk about earlier? It's that damage that occurs to those beta cells that leads to the rapid progression for some of these individuals, deteriorating the output of insulin, pushing them towards more of an insulin-dependent type state. So that's actually where you get your type 2s, that because they can no longer, it was just the whole insulin resistance issue for them, but because they've driven themselves to the state where they can no longer produce insulin, now you got a type 1 scenario, right? Now, I'll share something with you. One of the other tests that I do with patients is called a pre-DX test. And there's a scale on this test. It goes from 1 to 10, and it's a 5-year predictive algorithm for what their development of diabetes is going to be. The one correlation that I've noticed on this test that will throw that marker faster towards 10 than anything else is the C-reactive protein. That's our most sensitive inflammatory marker, right? That one marker will skew that test faster than any other marker on there. And you would think it's looking at blood sugar. So would glucose do it faster? Would HbA1c do it faster? Would insulin do it faster? No. It's the inflammation. The worse someone's inflammation is, the worse their progression towards dysglycemia is going to be. Right? And I'm kind of looking at some of you as I'm up here speaking. Some of you got dysglycemia in here. I'll share a little quick thing with you just related to this because, obviously we all have to sit there and listen to somebody else present for C's and all that stuff. I noticed that I tended to have the same issue when I got really diligent with my diet and really would watch what I was doing with my carbohydrate intake through lunch. I quit having issues, and I was always on with the speaker through the entire afternoon once I started doing that. So if you want to get the most out of when somebody's speaking, there's an idea for you. Fructose insensitivity. So really, when you look at this marker on the test, the thing I want you to translate that into is fructose intolerance. That's more or less what that means. And what we're trying to get at here is that the more fructose someone consumes, the more they're overloading this aldolase enzyme. That enzyme is only so efficient. It can only handle the metabolization of so much fructose at any one given time. That's why if you just bombard someone with it, you'll overwhelm this enzyme, and you'll get someone who runs into the production of things like the triglycerides and the uric acids. That's why I put this up here so you could draw the correlation on this when you look at these other labs with the micronutrient test, and you can really say, yes, this is exactly why these things are showing up. Fruit. Patients are funny sometimes, right? They'll say, well, I eat a good diet. Well, what do you eat? I eat fruit. Breakfast, lunch, and dinner, right? That's their plans. I mean, that's kind of the thought process on it sometimes, right? And yes, it is a good food, but it's got to be taken in moderation. You can't over-consume it. So even the consumption of good foods per se will cause this to happen. Now, let's switch gears for just a second here and go the other direction with our blood glucose. And I will tell you, and many of you have probably seen it too, what I'm getting ready to lay out for you as far as the pattern we're going to look at here, if I've seen it once, I've seen it 500 times. When you look at someone and you see that they've got a B5 deficiency, the first thing that should go through your head is, what is their steroid hormone production ability? How are they producing those steroid hormones? Do they have the ability to take that B5, convert it to acetyl-CoA, and then in turn convert that to cholesterol and follow through on the rest of the production of these hormones? And the two that you'll classically see in patients who have an impaired ability to follow through on this process, the first one is going to be cortisol, and the second one is going to be progesterone. Those are the two that are most generally impacted with this process. Now, that's the role that B5 plays in here. How does serine and vitamin C come into this? The way that serine comes in, your body takes phosphatidylserine and the memory centers up in the brain, your hippocampal region. Phosphatidylserine is used to resensitize those regions and to keep them as sensitive as possible, to keep your memory as good as possible, right? What happens if you've got someone who's in a chronic sympathetic state, they're producing cortisol on a repetitive basis. This is a feedback loop, right? So over time, when the cortisol feeds back up to the brain, it registers in the hypothalamus, it registers in the pituitary region. That area over time becomes blunted. It becomes less sensitive to it. And what you see is that as that occurs, is you get the overutilization of your serine to help maintain the integrity of that area of which that hippocampal memory center is tied directly in there. Okay? Your vitamin C. This was something I came across I was reading something out of the American Journal of Nutrition, and what it said is that every time the body secretes cortisol, it secretes vitamin C. And I got to thinking about that, I'm like, well, that makes sense, because what does vitamin C do? It maintains the function of the insulin receptors, it maintains the activity of those GLUT4 transporters. So what the body is doing is, it knows when it secretes cortisol as part of the sympathetic response, that cortisol has the ability to downregulate those insulin receptors. But if you secrete vitamin C at the same time, you can potentially offset the activity of the downregulation on those insulin receptors. You can actually take vitamin C, and someone who's running their glucose, and ask me later how to do this, but you can take someone who's running their glucose over 100 and use higher dosages of vitamin C and drive them back down into normal levels, into double digits, without driving them into a hypoglycemic state. And there's literature to back that up as well. So what are your primary treatment approaches going to be? Well, if we look at hypoglycemia, you're going to start with the B5. And the ranges on this are kind of broad in where you want to go. And it really depends on what your approach is. Whether you're an individual who looks at the micronutrient test and you think, okay, I've got a B5 deficiency, do I want to just replete B5? Or do I want to take a more synergistic approach with this and use the spectrum of nutrients that are playing into that state of dysfunction and address them as a whole? I tend to fall a little bit on the latter side, just because, and really what has transitioned me to doing that is looking at the micronutrient test. It's looking at when you typically see one of these deficiencies pop up, there will be other ones that pop up at the same time. So it's not as if you're just looking at a biotin deficiency by itself. You've got maybe a zinc deficiency tied in that, a B5 deficiency tied into that as well. Okay? But here's just some general recommendations for you. Your B5, 50 to 500 milligrams. Your phosphatidylserine, sometimes with this you'll hear that you should take this in the evening to help sleep or anything like that. I haven't seen that makes any difference. So, if you want to split the dose, do 100 in the morning, 100 in the evening, that's fine. That works just as well in my experience. Vitamin C, I guess it really depends on what line of thought you just prescribe to with your vitamin C thinking as well. I mean, I guess if you're, in the Linus Pauling camp, you're pretty good with going well above 5,000 milligrams, right? So, so really just wherever you want to go with that. But 600, I would say, is definitely a minimum there. And then what's really important? Food consumption. They got to eat on a routine basis. And I just put a general time frame in here of 2 to 3 hours. If you use 2 to 3 hours with someone and you don't see that that's getting it done, shorten it up. It may take going every hour and a half. I have one patient that cannot go more than an hour and a half right now. So, it's just, that's...
But what are they consuming though? Are they
Oh,
consuming carbs or?
No, it's a pretty
protein?
strict paleo diet.
Oh, okay.
Yeah, she's got a good diet.
Okay.
That's not the issue. And then the other thing that I didn't put in here that you definitely want to take into consideration is looking at those hormones. What is that state of cortisol? Do you need to get that cortisol back up? Right? Give the patient some help. I mean, yes, they've got to do the food component to this, but adding in the cortisol, helping them get their cortisol levels back up. And you don't have to directly use cortisol to do that. I mean, you can use pregnenolone, progesterone, whatever the case scenario dictates. But the bottom line is that overall, their 24-hour rhythm on their cortisol has got to come back up into a normal state. Now, your hyperglycemia, hyperinsulinemia side. Chromium, 400 to 600 micrograms is what you're pretty much thinking there. Yeah, your lipoic acid, 600 is definitely the low end on this. I topped out at 1200 here. You can go above that if you feel like you need to. If you've got a peripheral neuropathy, I would definitely say consider going above that. Your other thing